Okay. So for genus Haemophilus, this belongs to the family Pasteurella, and there are three important genera under this family. It's the genus Pasteurella, genus Actinobacillus, and then you have your Haemophilus. You will also notice that these three bacteria, they all cause respiratory disease, and most of these will cause pneumonia. No. So your Haemophilus, it is small, non-motile. It's a facultative anaerobe, and the morphology under the microscope is rods or coccobacilli. They do require one or both of the two defined growth factors that we will mention later. In the lab, we can grow this in blood agar or in chocolate agar. So they look like gray white colonies. They're very small. They're about 3 millimeters in diameter, and they produce a very thin zone of hemolysis. If we look at this image below, we can see some hemolysis on the blood agar. They can grow at 20 to 42 degrees, and on McConkey, they can also grow, but they will die within a week. They exhibit a characteristic known as satellitism, and this is with the help of a helper bacteria. For example, it could be Staphylococcus aureus. So we use the Staph aureus to make a cross, and the Haemophilus is growing dun sa quadrant, no? The bacteria, such as the helper bacteria, such as Staph aureus, will release two factors in the blood agar, which is your factor 10 and factor 5. It's hemin or and NAD, NADP, respectively. Then this these two factors, they can be present in chocolate agar as well. And this is prepared by using blood agar heated to 80 degrees, and the heat will release these two factors. So the bacteria will also grow here. Then this is Haemophilus on chocolate agar. They appear as very small colonies. In humans, we can see this. There is type B Haemophilus influenzae, and it causes the following diseases. There is non-typable H. influenzae as well, and these are the diseases that it causes. So the most important in humans, the species that's most important is your influenzae, parainfluenzae. So parainfluenzae causes pneumonia and endocarditis. There are also other species worth mentioning, Ducreyi and Aegyptus. But that's in humans. These are the ones that are important in animals. Most important is Parasuis and Gallinarum. Parasuis is a commensal of the porcine upper respiratory tract, but it does cause secondary bacterial infections when there is viral disease. So, yung Haemophilus parasuis, it is considered to contribute to porcine respiratory disease complex. And then we have Haemophilus gallinarum. This is in birds. It causes infectious coryza, and the disease is upper respiratory tract infection. And then in dogs, there is Haemoglobinophilus or H. canis, which is a fairly common commensal parasite of the lower canine genital tract. Then there are others, Paracuniculus and Aphrophilus, that are reported in other animals. The most important one that I mentioned is Haemophilus parasuis, which was recently renamed or reclassified to Glycerella parasuis. The disease caused is acute and has various combinations of meningoencephalitis, polyserositis, and polyarthritis. It is a significant contributor to bacterial pneumonia, or what is known as PRDC. I'll type this in. So I think you need to be aware of this disease because it is fairly common. So when we say PRDC, it is a combination of viral and bacterial diseases, and it can be a primary pathogen, but it can also be a secondary pathogen. So the viral diseases that are often seen in PRDC are PRRS or SIV, and there is another viral disease which is porcine circovirus or PCV2. So Glycerella parasuis was first isolated in 1931 in a pig with swine influenza. And so initially they didn't know that it was a uh, a bacteria. But in 1943, they recognized it as a bacterial agent and classified it under Haemophilus. The mortality is seen at any age and it affects healthy pigs as well. It's initially seen as fever, anorexia, and depression, and in severe cases, it can go into the brain, causing meningoencephalitis. And this will manifest as tremors, incoordination, paresis, and lateral recumbency. One characteristic of Glycerella is that it will produce these serous to fibrinous effusions on the serosal surfaces. And so we can see this yellowish uh, exudates on the peritoneum, pleura, pericardium, so on the heart, in the joints, and then in the meninges. So that's why we see the meningoencephalitis. So this is called polyserositis, and it is serous membrane inflammation with effusion, which is fibrinous. The clinical signs are not pathognomonic, but oftentimes when we see this, we associate it with Glycerella parasuis. However, you must differentiate it from E. coli, and so when it comes to E. coli, it can also cause systemic infection. Remember, there is extraintestinal E. coli and there's diarrheagenic E. coli. So extraintestinal can also cause systemic disease. But how can you differentiate it with Glycerella? So the fibrinopurulent meningitis is only found in Glycerella parasuis. Okay. Then for the isolation, it can be difficult because it needs the special conditions, the two factors for growth, but it can be attempted. This the isolation of the bacteria will allow for AST and serotyping, and that's also important because there are many serotypes for this bacteria. ELISA will detect the antibodies but cannot differentiate between serovars. So in swine practice or in swine production, the most common way that this is monitored is through ELISA or serology. And then PCR is used for difficult to cultivate organisms. And here we have an example of a detection of your H. parasuis. And yung positive band is seen at 800 BP. So this one is PCR, okay? There are some bacterians and they can be preventative. So there are commercially available bacterian vaccines based on Serovar 5, 4, and 5, or a combination of 1 and 6 for use in sows and pigs. And so the serotype, knowing the serotype is also important, but oftentimes this is not determined, no. And this can be successfully treated with antibiotics. And so here's a list of most of the antibiotics that are used. Then we can prevent the secondary bacterial infections by vaccinating with for the other viral diseases such as PRRS and PCV2. So if we prevent the viral infection, then we can also prevent the secondary bacterial infection. So that's a strategy. And then preventive treatments should be discouraged to avoid development of resistance to antimicrobials. So, okay. So any questions with regards to Glycerella? Okay. If there are no questions, yes, none. Would it be possible to request the recording? Yes, I already uploaded it. I just haven't shared the link. So siguro today I'll be able to share the link for the past recordings. Mm. Okay. Then so that's Haemophilus and Taylorella. So we're finished with these topics. So we'll move on to Pasteurella and Ornithobacterium, Bordetella, Moraxella, and Reimerella. I will try to finish this, or I will try to discuss as much as I can for today. And then next week, we'll finish off with these topics. So, siguro kasi next week mas konti yung topic. Baka yung iba dito, itong Moraxella Remirellia, maisama ko dito sa part na to. Okay. So, we will first um, discuss Pasteurella and Ornithobacterium. Okay. Um, Let me change it to Pasteurella. Okay. Since we just discussed Haemophilus, um, we will move on to another family member in family Pasteurellidae, which is your genus Pasteurella. So if you look at the family tree again, we have the family Pasteurellidae, and a member of this is your genus Pasteurella. So we're here. So we just finished Haemophilus, diba? So tandaan nyo, yung Pasteurelliaceae, sila yung mga respiratory disease. So it looks alike with, it looks very similar to your genus Haemophilus in that they are Coccobacillary and still gram-negative. They are non-motile facultative anaerobes. They are considered commensal parasites of animals. This means that they are commonly found in the respiratory tract of most animals. And here's an example of the uh, colony. They have circular umbonate elevation. That's Pasteurella. So remember yung umbonate, meron siyang knob dun sa ibabaw niya. The Pasteurella species possess capsules which contain acidic polysaccharides. They also contain hyaluronic acid, and as a gram-negative bacteria, it contains the LPS or the lipopolysaccharides. The capsule is very important to the pathogenicity of your Maltosida in particular, because the presence of the capsules allows this bacteria to um, cause it's a major virulence factor for your Pasteurella multocida. So it will cause 100% mortality at high and low doses. But um, this was shown in an experiment wherein the scientists or researchers uh, produced a mutant bacteria wherein they removed the capsule. They deleted the gene that produces the capsule on the bacteria. So here in figure A, you're seeing the carbohydrate-rich capsule that is on the outer part of the bacteria. And here in figure B, we are looking at the mutant bacteria, no, the capsule-deleted bacteria. And they found out that if you infect the animals or a test animal with your strain A, which has the capsule, then it will cause 100% mortality at high and low doses. And then your mutant or capsule deleted bacteria, it will cause 0% mortality at low doses. So this experiment tells us that the capsular proteins are very important virulence factors for your Pasteurella multocida. So apart from the capsule, we mentioned the LPS. The role of the LPS is to bind to the macrophages and cause direct toxicity to the respiratory endothelial cells. They also have the iron-regulated OMPs and toxic OMPs. This bacteria will have adhesins for attachment to the epithelial cells, leukotoxins as a defense mechanism. They will produce enzymes as well, which is the hyaluronidase and your neuraminidase. So your hyaluronidase, it will digest hyaluronic acid. So can anyone tell me where can we find, yun na lang, where can we find hyaluronic acid in the chat? 'Di ba yung mga mahilig um, mag, mag skin re, ah, s- skin regimen kasama yung hyaluronic acid sa mga dinalagay sa mukha. So where can you find the hyaluronic acid? Anyone? Okay, may mga sumagot. Skin, connective tissues. Okay, those are both correct. So it is found mostly in connective tissues. In the skin, it is mostly found in the dermis or in the dermal layer. And the hyaluronic acid, it acts as a cement. Parang siya yung nagbabind together nung skin tissues, no, and other connective tissues. And so if the bacteria has the ability to digest hyaluronic acid, then what it's doing is it's breaking down the connective tissue, right? And then at high concentrations, the leukotoxin has a cytotoxic effect, so it will kill the WBCs. And at low concentrations, it will have the ability to activate the leukocytes. So magkakaroon ng inflammation, no? So what happens is that so some bacteria, they have the ability to downregulate what is known as the MHC class 2 or the major histocompatibility complex. So the MHC class 2 is basically a way for the host cells to recognize that there is a foreign agent. And you'll learn more about this in immunology, but um, basically, when the MHC class 2 recognizes an antigen, it will trigger an immune response. And so if it has the ability to downregulate the MHC class 2, then it is a way for the bacteria to silence the immune system, at least for a while. Tapos, parang incognito si Pasteurella multocida. The bacteria produces what is known as a PMT or Pasteurella multocida toxin. This toxin is also known as the dermonecrotic toxin, osteolytic toxin, or the turbinate atrophy toxin. Mostly what it does is it controls the cellular actin cytoskeleton. So what happens is that it can go into the bone and it will lyse the bone. No. So mostly this is seen in atrophic rhinitis. So we'll try to see some pathology later so you can appreciate that. So Pasteurella are found in mucous membranes of susceptible hosts. In fowl cholera and hemorrhagic septicemia, transmission may be widespread. So these are two important diseases. Especially here in the Philippines, we definitely have this fowl cholera and hemorrhagic septicemia. This is in avians, obviously, and then hemorrhagic septicemia is seen in cattle or in carabaos. No, so here in Philippines, we had an epidemic of hemorrhagic septicemia where it killed a very high population of our carabaos, no, Philippine carabao, and so this is an important disease, no, here in the Philippines. Infection may occur through inhalation, ingestion, or direct contact with open wounds, and many infections are endogenous. So, if you do you remember or do you know the meaning of endogenous? So, what is an endogenous infection? Can anyone tell me in the chat? What do we mean by endogenous? A lot of the infections that we see are exogenous. So, ibig sabihin, nakuha natin sa labas, di ba? Or we got it from other people, or it was transmitted. But when we say endogenous, what does that mean? Okay, so the infectious, sabi ni Nicole, the infectious agent causing the disease is already present in the host. Yeah. Yes, that's correct. So, since this bacteria is a commensal, then in some instances, when an animal is infected with the bacteria, then it was already there, no? So there may have been triggers, for example, stress or secondary or viral infections, but essentially, the bacteria is already present in the host. Okay. The severity of lesions depends on the site of infection, the virulence of the strain, and the resistance of the host. And vascular damage results in hemorrhage and fluid loss. There may be focal necrosis in parenchymatous organs and ulcerations on mucous membranes. During pneumonia, inflammatory cells infiltrate the lung parenchyma with erythrocytes, neutrophils, and mononuclear cells. So there is a very strong inflammatory response that can be seen in Pasteurella infection. The lung tissues will appear black to red, pink, and gray, and there may be necrosis, abscess formation, and fibrin deposition in severe cases. In other parts of the body, the bacteria can also spread through the joints, the ears, ovaries, wattles of birds. This is in chronic fowl cholera. And then there can be disturbed osteogenesis in the snout, in particular the turbinates and bones of the snout. And so this is seen in atrophic rhinitis. So if we look at the different animals, they have each of them has a particular disease, and some species that are recognized to cause these diseases. And then we have the symptoms here. So we have, for example, in cattle, I mentioned this, there is hemorrhagic septicemia, and it's caused by your P. multocida. Then there is another disease which is called shipping fever. So shipping fever is not only caused by Pasteurella, but it is one of the causative agents. And then in when this happens when there is a very stressful event in the life of the animal. So for example, during transport, this usually occurs kaya yung tawag sa kanya is shipping fever. And then what happens is there is high fever, diarrhea, inappetence, and some respiratory signs. In sheep and goats, there is septicemic pasteurellosis and enzootic pneumonia. So this can be caused by Trehalosi and your hemolytica. And then some symptoms or some diseases could show as gangrenous mastitis. In swine, we have fibrinous pneumonia and atrophic rhinitis. So this is caused by your multocida. So the symptoms are sneezing, epistaxis, staining of the face, tear duct obstruction, and lateral deviation of the snout. Then in rabbits, there is snuffles. This causes bronchopneumonia, middle and inner ear infection, conjunctivitis, septicemia, orchitis, melanogastritis, and pyometra. In birds, we have fowl cholera. This causes anorexia, diarrhea, nasal, and ocular discharge for acute cases. In subacute cases, there is rales and mucopurulent discharge, and in chronic cases, there is gaseous lesions. In dogs and cats, Pasteurella can cause wound infections, pyothorax, and foreign body lesions. Mostly there are purulent exudates. Then in horses, there is equine respiratory disease. The species associated is Pasteurella caballi, together with another bacteria, which is your Streptococcus zoopidemicus. In small lab rodents, Pasteurella is recognized as well. It causes pneumonia. The causative agent is your pneumotropica, which is considered as a commensal. And then in humans, um, it can cause wound infections from animals. So here are some examples of the lesions associated with your Pasteurella multocida. This is your swine, and this can cause chronic bacterial pneumonia or fibrinous pneumonia. You will notice that the both the cranial and caudal lobes are affected by this bacteria. So kadalasan, no, even the deeper parts of the lung are affected. It's not just the upper respiratory tract. And then this is a cross section. Can anyone tell me what these are in the chat? Can you recognize what this is? Yes, this is the nasal cavity or the snout. These are turbinates, and these turbinates, they become lysed, no? So there is the osteolytic toxin that is produced by your Pasteurella. And what you'll notice is that this is asymmetrical. So there is damage on both sides, I think, but more pronounced on this side. So on the left side. And in it, if you look at this, wait, not sure if I have the slide here, pero yeah. Okay. So if you look at this na hindi pa na cross section, no, this is what it looks like. This is atrophic rhinitis. There is the lateral deviation of the snout. And this is because of the osteolytic toxin produced by your Pasteurella. And please take note then the atrophic rhinitis, Pasteurella multocidi is only one of the bacteria. No? There's another bacteria involved, which we'll discuss later. And then this is blue bag. So this is mastitis in your sheep, also caused by your Pasteurella multocidi. So Pasteurella adjuvant bacterins are protective against hemorrhagic septicemia for 2 years. So here in the Philippines, we are required to vaccinate the cattle and our carabaos with the Pasteurella bacterin to protect against hemorrhagic septicemia. And then there are some vaccines for fowl cholera, so Pasteurella din yun. Then inconsistent benefits from most shipping fever vaccines, right? So yung sa shipping fever, yung sa cattle, hindi masyado effective, mostly because in the shipping fever, there are other bacteria and viruses that are involved. So for example, there is bovine herpes virus 1, there is mycoplasma, etc. So these other um, causes of shipping fever, um, it makes it very difficult to produce a vaccine. No? Kasi multifactorial siya. No? So when we say it's a multifactorial, there are other causative agents that will play a role in the development of that disease. Then we have bacterins with toxoids against Maltosida and Bronchiceptica. So, ito yung sa atrophic rhinitis, no? So, atrophic rhinitis is caused by P. Maltosida and Bordetella bronchiceptica, and most of these are effective against atrophic rhinitis. Yan yung dun um, sa lateral deviation of the snout. For the diagnosis, it can be through the isolation of the bacteria. We can use the exudates, tissue impressions, sediments of transtracheal aspirates, and in blood smears, if there is septicemia, we can see the bacteria. Here's an example of hemorrhagic septicemia, blood smear, where there is the bacteria in the presence of the blood. They will have a characteristic bipolar staining, and then on culture, we can grow them on BAP. This bacteria has a moderate resistance to aminoglycosides, but most are susceptible to antimicrobials. For food animals, it's important to consider the withdrawal period. And this is always emphasized, especially in poultry or in swine production. If you're giving antibiotics, you need to practice the withdrawal period, meaning that you have to allow a certain time to pass before you slaughter the animal after giving the antibiotic, because the antibiotic can remain in the tissues, no, or in the muscles. So we don't want um to have the residues, right, in the meat, especially. Here are some of the antibiotics that are used for Pasteurella. They are commonly administered through the feed and water as prophylaxis or therapeutic. Then confirm appropriateness of antimicrobial using the susceptibility test. This is important so we don't contribute to the AMR. And then immunization is effective for Hemocep and atrophic rhinitis. Okay. Then most strains are susceptible to penicillin. So here's a AST of P. maltosida, um, and uh, showing its susceptibility to penicillin. So here we see the zone of inhibition. And this is uncommon for gram-negative bacteria, but for Pasteurella multocida, this is common. So in summary, this bacteria is a facultative anaerobe and commensal of the respiratory tract. That's why most infections are endogenous. The capsule is very important for this bacteria as a virulence factor, and it can cause respiratory and systemic disease in domestic animals. Diseases may be triggered by stress and co-infections. A very good example of this is the shipping fever. Okay. Any questions with regards to Pasteurella? So that's clear. Okay, we will move on to your Ornithobacterium. For Ornithobacterium, there is a single species that is important, which is your Rhinotracheale. So Rhinotracheale, they appear as pleomorphic. So in here we see the gram stain. They appear as elongated, and some are small pieces, no, or some small rods. This is a relatively recent bacteria, kasi it was only discovered in 1991. Were you born in 1991? Pinanganak na kayo nun? Hindi pa? Or some of you maybe. Raise your hand if you were born, kung buhay na kayo in 1991. Wala. Wala nga dito na born in 1991. 1999. Wow, okay. In 1991, grade 1 ako nun. So, dun pa lang na-discover si Ornithobacterium rhinotracheale. Oh, yes. Okay, so this is a Pasteurella-like bacterium, kaya tinidiscuss natin siya together with the Pasteurella. It's a slow-growing bacteria with many, many strains and serotypes, no? It's an important bacteria kasi it causes these respiratory infections that can lead to massive economic losses in the poultry industry. So, yung disease, it causes air saculitis and pneumonia. It can be a primary or secondary etiological agent, and they can cause growth retardation, and the carcasses are unacceptable for human consumption. The disease was first described in South Africa by John Dupreez. This was in broiler chickens. And relatively mild respiratory symptoms, which start with sneezing at the age of 28 days and can last up to the end of the fattening period. Then there can be mortality, about 3% or more, and poor performance. For example, growth per day and feed conversion is decreased. So at post-mortem investigation, there can be the yogurt-like exudate in the air sacs, and mostly this is seen in the um, abdominal air sac. And there can be pneumonia. The most important clinical sign is decreased feed and water intake. However, please take note that in any disease in birds, maybe also in other species, one of the most one of the earliest signs of disease is decreased feed and water intake. No? So this is not specific to ORT. This can also be seen in other viral and bacterial infections. Then there's growth retardation, weakness, dyspnea, mucus discharge, mortality, and different strains are equally able to infect other avian species. So mostly this is the pathology. We mentioned that earlier, and this is what it looks like. So kanina sabi, di ba, merong yogurt-like exudates in the abdomen or in the abdominal cavity. And um, the this is the fibrin clots that were being described earlier. So there is erysaculitis, and this is in a 28-day-old broiler. Then we also see the pneumonia. Here, what's being described is that there is a very distinct border by which we can still see some healthy part of the avian lung. So this part, and then this part, there is lesions associated with pneumonia. So there is hemorrhage. There is also uh, probably consolidation, no, and mostly inflammation of the lung. The bacteria will be found on the respiratory epithelium. So if you look at this picture, the brown spots, these are immunohistochemistry uh, positive for ORT or Ornithobacterium rhinotracheale. So initially, nandun siya sa epithelium, no? And then it will invade the lung tissue over time or the respiratory tract. So after a few days, no, 4 days post-exposure, the bacteria will invade the epithelium and then go into the submucosa. Eventually, it will cause damage to the respiratory tract. No? So the brown spots are the positive for ORT. So mostly these are yung progress this slide shows the progression of the invasion of the bacteria. When oral antibiotics perform poorly, we can use injectable tetracycline and penicillin. Some strains slightly sensitive to enrofloxacin and TMPS. However, some strains are resistant as well. Okay. Any questions with regards to ORT? Okay. Um, so we have a little bit more time. Baka naman pwede pa. Kaya nyo pa? Go lang or tama na? Tama na, Doc. Ayaw nyo na? Kaya pa? Sige. Okay, one more. One more topic para hindi tayo mahuli. Catch up lang tayo. Okay. Kasi lahat ng ito, you might think na hindi, ano, um, baka hindi naman itanong ni Doc sa exam or hindi naman importante, but pwedeng lumabas siya sa board exam. So if it's not me who will ask the question, it's the board examiners that will ask the question. So hopefully you could still recall this and at least nasa inyo yung material when you graduate and review for the boards. Okay. Let's go to Bordetella. So kanina na-mention natin yung atrophic rhinitis. And this is usually seen in pigs. So itong Bordetella bronchiceptica, it's very morphologically similar to your Pasteurella. Ano. So ganun din siya, no? Gram-negative Coccobacillari or Coccobacilli. And this was named after the Belgian biologist Jules Bordet. And he lived in 1870 to 1961. He first isolated another species, which is in humans, the pertussis, no, Bordetella pertussis. They are considered as parasites of the ciliated respiratory epithelium and has capsule-like envelope. This is seen in Bronchiceptica. Bronchiceptica also has villi and are considered as motile together with B. avium. They release the dermonecrotic toxin, which we mentioned earlier. It's just like the PMT, the Pasteurella multitoxida toxin. And then we have other virulence factors that are very similar to other uh, bacteria that we have described in the past. No? So yung adhesion niya, this binds to the sialic acid residues on the ciliated epithelia. Kaya yun yung manner niya of attachment to the cell. The Bordetella bronchiceptica produces a hydroxymatic siderophore, which acquires iron from iron-binding proteins of the host. Kaya um, nagkakaroon ng iron deficiency din. So, yung siderophore for B. bronchiceptica, it's an important virulence factor as well. So here are some of the virulence factors in Bordetella pertussis. We have the Fimbriae, outer membrane proteins, LPS. I'm sure you're familiar with this kasi describe na natin to sa E. coli or sa mga Enterobacteria siya. Then we have the hemagglutinin, the pertussis toxin, heat labile toxin, and adenylate cyclase toxin. The bacteria is a strict aerobe, so you'll notice that a lot of the lesions and diseases that it will cause is only restricted to the upper respiratory tract. Kasi kailangan niya ng oxygen. Hindi siya makapunta dun sa deeper parts of the lung, unlike your Pasteurella multocida, no? So, napansin niyo yung sa Pasteurella, yung lesions niya, umaabot dun sa deeper parts of the lung. Kahit na low on oxygen yung area na yun, pwede mag-survive yung Pasteurella. But in your Bordetella, it will be restricted to the upper respiratory tract because it's a strict aerobe. And then it utilizes amino acids, grows in McConkey, so pwede siyang ma-culture in the lab. Pwede rin siya sa blood agar. and it can grow intracellularly or inside of the cell. This bacteria does not ferment carbohydrates and utilizes citrate as an organic carbon source. So, it will undergo protein metabolism, no? So, napansin nyo yan, pattern din yan na common. So, yung mga strict aerobe, they do not ferment carbohydrates. Instead, they will um, undergo protein metabolism. Naka-keto diet sila. Okay, and these are colonies of your Bordetella bronchiceptica on bovine blood agar. And here we can see yung colonies, no, white, round, shiny colonies. They can be found in the ciliated respiratory tract, mostly seen in wild animals and domestic carnivores, rodents. Yung pinaka-common na disease is seen in swine. And then we have other animals, bee avium, mostly found in fowls and turkeys. This is a bacteria that's frequently isolated from respiratory infections of multiple etiologic factors. Kaya parang nakikisali siya dun sa secondary bacterial infection. No? Then transmission can be airborne or via the water and litter. So what happens, so the pathogenesis is that the bacteria will attach to the ciliated respiratory epithelium. It will proliferate and it will cause cilial paralysis. Eventually, there will be the development of inflammation, nasal irritation, and the turbinates eventually will become susceptible to the dermonecrotic toxin of your Pasteurella mutacida. Here are some lesions of Bordetella infection. You will notice that with Bordetella alone, so this is infection with Bordetella alone, most of the lesions are confined to the cranial parts, cranial lobes of the lung. No? And here is another one where we see the cranial lobes affected. However, if we infect this animal with other bacteria or viruses, I think in this experiment it was PCV2 if I'm not mistaken, or PRCV. Okay. Or co-infected. Hold on. Yeah. PRCV or co-infected. Okay. So here is coronavirus, no? And then so on the left, virus only, and then on the right, it is co-infected with with Bordetella bronchiceptica. So here we see the lesions are more severe, and we can see that the ventral parts of the lung are also affected. And then in letter C, um, these cross lesions are ganun ulit. No? Okay. So on the left, there will be this is infected only with the porcine coronavirus, and then on the right, co-infected with Bordetella bronchiceptica. And here we see more severe lesions, diba? Then there is histological grading for rhinitis. This is normal ciliated epithelium. We can see the cilia right here. All of these cells are intact. And then when it is infected with your Bordetella bronchiceptica, then we can see that the ciliated epithelium slowly becomes uh, atrophic or um, actually necrotic, no? So they die because of the infection. So uh, as the disease progresses, um, the ciliated epithelium becomes eroded, and that's when we see the high grade rhinitis, no? And in pigs, this will cause AR or atrophic rhinitis. We described this kanina na sa Pasteurella. So there's turbinate atrophy, disturbed osteoblast physiology. In dogs, there is a disease called canine infectious tracheobronchitis. What is known as also it is this is known as kennel cough. This will produce mucoid mucopurulent exudate in the lungs, and lymph structures are also affected. This disease is important in multiple um, pet households. For example, in breeders or in kennels, diba? Where there are many animals, many dogs being kept in a confined space, then this can become a problem. It may be difficult to treat. Then there are also vaccines available. In cats, there is mild upper respiratory tract infection, sneezing, and coughing. Then in turkeys, this can cause acute coryza. The local antibodies prevent colonization of lungs in dogs, and provide we can provide bacterins to pregnant sows. Then it can provide colostral immunity. Bacterin toxoid preparations for piglets. For the atrophic rhinitis, this is available. Then for dogs, there is live attenuated vaccine for kennel cough. There are some people or some studies then siguro that advocate intratracheal administration of the live attenuated vaccine, mostly because you want the local affected areas to be exposed to the antigen para magkaroon ng local antibodies. Um, but hindi yan yung conventional way of administering this vaccine. Okay? Just be aware. And then Bordetella avium, meron ding live attenuated vaccine. For the diagnosis, we can use nasal swabs and transtracheal washes for culture in the McConkey agar. There is no treatment for the atrophic rhinitis, only preventive measures. Then prophylactic use of sulfonamides can be given in feed and water. For kennel cough, I mentioned this earlier, it's difficult to treat in dogs and it does not respond well to antibiotics. The avium is susceptible to tetracycline, erythromycin, and nitrofurantoin. Okay, and that's it for Bordetella. So I think I will stop here. If you have any questions, feel free to send me a message sa chat. Then um, kindly await yung mga further announcements for our new exam schedule. Um, and I hope everyone is safe. And siguro, if you're still in Los Baños, you stock up on food and water. And hopefully, we'll get through this, no? Sana hindi masyadong malaki yung damage sa Los Baños, no? Kasi it's really devastating pagka may ganitong bagyo. Um, Maraming issues din kami sa building, nawawalan ng kuryente, yung mga ref, nawawalan, nasisira yung mga gamit, no? So, um, hopefully, hindi masyadong affected ng LB. Okay. So, I'll see you guys next week, hopefully.